So now smell and taste. Allow me just to mention something so that we don't come on it when we see the question when it comes to smell and taste. Not much talking about this because you already know, but then how does the transduction, because it can also come as an essay, how do you really perceive smell? So in our nose, we know we have got the olfactory epithelium that has got different types of cells. And what's happening is that we have got Bowman's gland, which releases the olfactory binding protein, which concentrates the odor. And so what will happen is that when this odorant attaches to the receptors, what it does is it is going to activate. Hope the odorant is here. You can see it is attaching to a G-coupled protein, and there are two mechanisms by which you are going to generate an action potential that is going to lead to the information going to the central nervous system via cyclic adenosine monophosphate or inositol triphosphate IP3. So the first mechanism, when the odorant binds to the receptor here, what is going to happen is that you have formed the odorant receptor complex, and that is going to activate the G protein. The G protein is initially bound to GDP. That GDP will move and then GTP will bind. And then what that is going to happen is that now the G protein with the GTP is going to activate adenylate cyclase or adenylyl cyclase. And that guy, what it does is that it converts ATP to CAMP. And when you form your CMP, it's going to go and bind to the sodium and the calcium channels, thereby opening them. When you open these guys, what happens that there's an influx of positive charge, and that is going to lead to depolarizing generator potentials. That is the first mechanism. Another mechanism is that when the odorant had to bind to the GTP, to the G protein, and then caused the binding of GTP to the G protein, instead of that guy going to activate adenylate cyclase, you can as well activate phospholipase C, and that phospholipase C can form IP3. IP3 can still activate the calcium channels, and that will still cause the polarizing generator potential. And at the end, because we have got this depolarizing generator potential in both mechanisms, because it's leading to calcium influx or an increase in concentration of calcium, that increased calcium concentration is going to open what is known as calcium gated chloride channels. And that will cause the chlorides to start leaving the cell, and that just leads even more to depolarization. When the depolarization reaches the high local, the axon, it will generate a true action potential when you reach threshold, and then information is going to be sent to the central olfactory pathway. And you can see for the central olfactory pathway, you have got important cells here like the mitral cells and the tufted cells. These guys, you can see there's going to be information going via the olfactory tract, and it's important to know where they are going. You're having the medial olfactory tract, which is going to the basal forebrain limbic structures. You're having also the lateral olfactory tract. But of importance is that you are going to the olfactory cortex, and then to the thalamus, and then to the prefrontal cortex. Some of them are going to the amygdala, and then to the entorhinal cortex, still going to the hippocampus. You can see that smell is really related to the limbic system. That's why when you smell something, you feel, remember, like the hippocampus is involved in memory. So you know some smells of people. Think of the smell of your boyfriend or your girlfriend. Even when you've not heard their voice, if they just pass, even when you're sleeping, you can even wake up. My girlfriend or my boyfriend has come because of that information. And then information is also going to the amygdala. You know, the amygdala is the emotional center. When you smell some things, you are feeling nice. Like, for example, when you're in the restaurant, you even want to cry because you smelled chicken, right? It's now emotional. You want to cry, you are too hungry, you have smelled the chicken, and you feel too nice. That's amygdala. For taste, the only thing that I want to mention is that taste sensations are picked at different regions, like the back of the tongue, bitterness. the front, the tip, the sweetness, and then on the sides you can see the saltness and the sour, the SS. But this doesn't mean they are different receptors which are picking up these sensations. The only difference is the concentration of the receptors in these different regions. So when it comes to acid, this acid which is in the side or the sour, so for you to generate an action potential for the sour or the acid, what happens is that action potentials are generated via potassium voltage gated channels, and hydrogen plays an important role in opening those potassium gated channels. For the salt, which is also on the opposite, for the salt, it's the sodium channels. There's an influx of sodium. And then when you come to the, let's start with the sweet, which is in front. For the sweet, there are two mechanisms. They can either be influx of potassium, and that causes depolarization, or there is a G protein. So what happens is that when the G protein is activated, there is now going to be the action potential being generated like we discussed for the others, G protein. And then for the, oh, actually what I'm from talking about is the beta, where you have got if influx of potassium and also the G protein. For sweet, it is also a G protein, but the only thing is that adenylate cyclase is going to produce CAMP. The CAMP can do two things. It can either activate phosphokinase, which is going to lead to closure of potassium channels. When you close potassium channels, what does that cause? Potassium will not be leaving the cell, so there's accumulation of positive charge that will lead to depolarizing generator potential. Another thing, instead of CMP activating phosphokinase, it can activate phospholipase C, which produces IP3, which opens calcium channels. Allow me just to mention on the beta that the G proteins for the beta test, what they do is that they close potassium channels, just like on the sweet. When you close the potassium channels, potassium is not leaving the cell, and so there is that generation of the action potential. So where the fibers go for taste, you're having three nerves. Cranial nerve number 7, which is the facial nerve, taking information from the anterior two-thirds, and then from the posterior tooth, anterior one, anterior one two-third, and then posterior one-third, it is the glossopharyngeal, and then the epiglottis, you are having the vagus. This information is going to go to the ventral-posterior medial nucleus of the thalamus. Please take note, ventral-posterior medial, and then it will now go to the gustatory area. But then what happens at the information, the first order neuron is going to go to the solitary nucleus or the nucleus solitarius, and then the second order neurons are going to go to the ventral posterior medial nucleus or the thalamus. The third order neurons are now going to go to the test area 43, which is going to be found in the anterior insula and the frontal opercular. Hope we know the meaning of opercular. Remember the insular cortex, those regions of the frontal lobe, the parietal lobe, and also the temporal lobe that covers the insula. We call that as the opercular. Okay, basically that is what I wanted to mention. So you can start here. Relay station for tests are: A, accessory nucleus; B, inferior nucleus; C, nucleus structure solitarius; D, superior colliculi. E, all are correct. Anyone? The nucleus structure solitariums. Thank you. The olfactory bulb. Olfactory bulb. A, projects to sensory cortex through V, A, N, A, or the nucleus structus solitarius. Yes. And then B, the olfactory bulb. A, projects to the sensory cortex through the ventral anterior nucleus of the thalamus. ventro-anterior nucleus. The A or there you can guess what is meaning. And then B projects to sensory cortex through the ventro-posterior lateral nucleus of the thalamus. Maybe those PMOs are representing other things. Projects to the solitary tract so that smell and taste can be combined. D receives a cholinergic innervation from the nucleus of the diagonal band, shares a type of myelin with the oculomotor nerve. Anyone that can help? Pardon? Or C projects to the solitary tract so that smell and taste can be combined. Is there anyone with a different option? Okay, good. Which of the following statements regarding the transduction of the taste stimulus is correct when a chemical molecule interacts with a specific membrane site on the microvilli? So basically everything here is true. Which of the following is true about the olfactory receptor? They are located in the olfactory epithelium in the roof of the nasal cavity. They are modified bipolar neurons that can regenerate. They express only one type of olfactory receptor, protein out of about 1,000 of the above. Anyone? Okay, the answer is all of the above. Do you actually know that we have got different types of receptors for smell? Each receptor is supposed to respond like to an odorant, but of course there can be those interactions. Do you know that the neurons or the genes that code for smell, for these smell, are actually the most numerous in the body? That is because we have got so much different types of smells. And do you know for animals, females can be attracted to males using what is known as the pheromones. So maybe this is why we love going after ladies. Maybe they produce pheromones and we smell that, right? But I don't know about that in human unless in animals. There is a contribution from anyone? A question? Okay. Which of the following is a mechanism of adaptation to prolonged odorant stimulation? Why is it that you step in a room where, yes, doc, all of the above? Mm. So imagine you go in a room where your special person is cooking chicken. When you stay in that room for like 10 minutes, 15, 20, 40, you stop smelling the chicken. So you have adapted. So why? What causes that? Increase the synthesis of cyclic adenosine monophosphate, increased opening of sodium and calcium channels in the olfactory receptor cells, decrease the release of neurotransmitter from the olfactory receptor cell, or of the above. Anyone? Decrease release of neurotransmitter from the olfactory receptor cell. Okay. Any other option? Maybe there can be more than that. Is there anyone with another option? We all agree with D so that we move on. Pardon? Okay. So, just so that we can be aware, some things that can lead to this desensitization or the adaptation. Sometimes the odorant, when it gets to bind to the receptor and is attached there, like it's still there, the receptor will become less responsive. Sometimes also there can be a downregulation of the expression of the receptors, or even some neurons which are inhibitory in nature. So inhibitory neurons and also the cortex can as well come in. And also there's just where what's known as habituation. So those can get to forming adaptation. But we can still pick the what did someone say? They said D, isn't it? Or I didn't get well. All right. Which of the following is true about the gustatory receptor? They are located in the taste buds, on the tongue, and other parts of the oral cavity. They are specialized epithelial cells that release neurotransmitter onto the afferent nerve fiber. They can respond to more than one type of taste testant molecule of the above. Thank you. I think I also go with this one. Which of the following testants is detected by a G protein coupled receptor mechanism? Sodium ions, hydrogen ions, sweet molecules, all of the above. Stay sweet, right? Yes. Which of the following cranial nerves carries taste information from the anterior two thirds of the tongue? Facial, glossopharyngeal, vagus, trigeminal. The facial. Thank you. And that's where these questions end.